Midterm examination of fish genetics. Number 1 question. Enumerate the modes of gene actions and determine the phenotype and genotype of the progenies produced in each mode. Enumerates the modes of gene action and determine the phenotype of the progenies produced in each mode. There are 6 modes of gene action. Number 1, recessive epitasis. Number 2, dominant epistasis. Number 3, dominant and recessive epistasis. 4, duplicate recessive epistasis. Number 5, duplicate dominant epistasis. Number 6, polymeric gene interaction. Number 2, differentiate dominant from recessive gene. Dominant traits are always expressed when the connected allele is dominant, even if the only one copy of the dominant trait exists. Recessive traits are expressed only if both the connected alleles are recessive. Number 3. A dominant G allele in common CAR produces normal pigmentation and the recessive G allele produces gold pigmentation. A. What are the genotypes of the parents? B. What are the genotypes of progenies? And number C. What genotype should be mated to produce an old phenotype? Number 4. Based on the theory discussed of dihybrid cross, list all the gametes produced by the dihybrid cross of two homozygous gray guppies with curved spine. And letter B. Draw a Panel square and determine the genotype and phenotype ratios expected from the cross. Number 5. Based on the given example on X-linked genes, how are transplanted tailed guppies produced? Draw a Punnett square and determine the phenotype-genotype ratios of progenies and cross between the mating of heterozoans caudalus female and transparent tail male. Mutation, the changes in the sequence of genes in DNA, are one of source genetic variation, another source in gene flow, or the movement of genes between different groups or organisms. Final genetic variation can be result of sexual reproduction. Number 7. Understand and apply the concept in selection in fish stock management. A stock assessment is the process of collecting, analysis, and reporting demographic information to determine changes in the abundance of fishery stock in response to fishing and to the extent possible. Number 8. What is genetic drift and how can it be prevented? Genetic drift is the change in frequency of an existing gene variant of the population due to a random chance. Genetic drift may cause gene variants to disappear completely and may thereby reduce genetic variation. It could be caused initially where alleles to become much more frequent, and even fixed genetic drift is technically not preventable. However, it can be limited by two factors. First, the population in question can be significantly large enough to accommodate random factors in breeding.